So today we're going to talk about vision, and I just want to be clear up front that this is not the Marvel character. So if you came to this lecture thinking that we were going to talk for an hour about the Marvel character Vision, that is incorrect. I'm very sorry. So when it comes to vision, it's partially dependent on how far light can travel. Now we know that light can travel very far because we get light from the sun, but let's just say there's something that's like impeding your vision. There's your eye right there, right? If it's blocking that, then you're not going to be able to see. Now, when it comes to vision, we talk about the eyes a lot, but that gives us the sensory information that our brains then compile in order for us to see things, right? If you think about it, colors don't actually exist. They are just wavelengths that our brain assigns meaning to. That's our brain. So the perception of vision, not the sensation, but the perception is in the brain. So we've talked about different types of neurons. You might remember the, sensory neurons. They have specialized receptors that are sensitive to particular types of energy. So some are responsive to sound, some are responsive to temperature, etc., etc. The law of specific nerve energies states that activity by a particular nerve always conveys the same type of information to the brain. So our eyes aren't going to start being responsive to sound. They're always going to send the same impulse, which tells the brain, we're perceiving light. So light enters the eye through the center of the iris, which is called the pupil. We have a lens which focuses it with the cornea onto the inside surface of the eye, which is the retina. And that is lined with visual receptors, rods and cones. We'll talk about that later. Just so you know, the left side of the world strikes the right side of the retina and vice versa. So it's actually switched. Same with the top and bottom. So the things from below, like things that are on the floor, are going to hit the top of your retina. Things that are on the ceiling are going to hit the bottom of your retina. So we have bipolar cells, which are located closer to the center of the eye, and they receive messages from the visual receptors at the back of the eye. Those cells send messages to a bunch of ganglion cells that are even closer to the center. And then those axons will join to form the optic nerve that travels to the brain. You might remember from previous slides the optic chiasm. So if you remember seeing that and going like, what's that? That's related. So that will come up in a bit. Then we have amacrine cells, which receive information from bipolar cells and send it to other bipolar ganglion or amacrine cells. These control the ability of ganglion cells to respond to shapes, movements, and other specific aspects of visual stimuli. So if you see this square moving quickly, that would be your amacrine cells responding not only to the shape, but also to the movement. So here's a cross section of a vertebrae eye. So imagine you're looking forward, right? So you have the pupil here, the cornea, and the lens, right? You have this muscle which helps squish it so that you can focus your lens. Then squish it is the proper medical term, just in case you're wondering. Then we have our rods and cones, which are responsive to specific wavelengths of light. Then you'll notice the blind spot. So there's actually a part in the retina where there's a little hole because the optic nerve has to come through, right? So there's in your field of vision, there's this little hole, and you actually aren't getting any visual information in that area. You might be wondering, well, how do I see? Well, two things. You have, well, in most cases, you'll have two eyes, right? So you're able to fill in that information, but also your brain can make assumptions about what's in the world, right? So just by existing, your brain will go like, okay, well, if there's this line that I see, I can assume that that line is continuous. So here's the visual path within the eye. So you can see the different layers of cells. So the receptors, the rods and cones, will send their messages to bipolar and horizontal cells. Those cells will send messages to the amacrine and ganglion cells, and then the axons of the ganglion cells form the optic nerve, which is this big old thing right here, which exits the eye and sends the message to the brain. So, as mentioned before, the optic nerve leaves the eye. It's that bundle of the axons of ganglion cells. Uh, because it's leaving the back of the eye, there is a blind spot right at the point of exit because there are no receptors. So here's a little illustration of the blind spot. Uh, Hopefully, the screen that you're on allows you to do this. If not, just you know, you can imagine, or maybe you can't imagine, and you'll always wonder what you missed out on. So sad. Here are the instructions. So close your left eye and focus your right eye on the O in the top part. Move the screen towards you and away, or move towards or away it, and notice what happens to the X. At a distance of about 10-ish inches, the X should disappear. Now try it again with the bottom part. At that same distance, what do you say? Does that change your perception of it at all? This is showing you how your brain misses information, but also how your brain might fill in information. So the fovea is the central portion of your retina, and it allows for very detailed vision because it is packed tightly with receptors, and it's nearly free of ganglion axons and blood vessels. So it's just focused on being focused. I hated saying that, but what are you gonna do? It's focused on being focused. So I mentioned rods and cones before. We'll go into detail about the differences between those two a little bit later. But the fovea is almost exclusively composed of cones, and those receptors attach to a single bipolar cell and a single ganglion cell, known as a midget ganglion cell. Is that a term that we should still be using? Probably not. That is, I forgot that that was a term. So yeah, we'll we'll need to fix that. We, the entire scientific community. So each cone in the fovea has a direct line to the brain, which allows the registering of the exact location of input. Again, these are because it's so focused on focus. You want to give each receptor the ability to get that information directly to the brain. And our vision is primarily dominated by what we see in the fovea. think about like where you're looking right now, right? The periphery, the things that are outside. So if you're looking at the screen, right, that's the thing that's most important. And even though there are things in your periphery outside of that circle of focus, those things aren't as important. If you're reading, you're focused on the words right in front of you. The words around you, it's good to know that they're there, right? But you don't need to precisely focus on them. Now, if you see movement outside of your phobia, then you shift your entire attention and focus on that movement, and then you see that there's a moth flying around your room, right? So wherever our phobia is focused, that's where we're going to see the most detail. So when it comes to birds, I'm not sure if you knew this, but birds fly. So they are used to their prey being below them, meaning the top half of their retina are more sensitive to detail. So if an owl needs to see something above itself in detail, it's going to actually turn its head, right? So the reason for that is because it needs to be able to get information from here to the top of its retina. So in the periphery of the retina, so everything that's not the fovea. Did I switch my pronunciation of that? Phobia? Phobia? Fovea? No, it's fine. Just just use both. So the periphery, there are a greater number of receptors, mostly rods, and those converge into ganglion and bipolar cells. Uh, you have less detailed vision, but you're much more able to perceive things in lower light in that periphery. So here we have the cones in the fovea, and we have the rods in the periphery. So you can see here that basically you have one cone per bipolar cell, uh, but you're also receiving inhibitory input from surrounding cells. Here, you only have two cells with a bunch of rods. So you're going to get, because you're getting input from lots of rods, even if there's less light, you're more likely to get visual information as opposed to the cones where you really are going to need strong enough visual information for this signal to be triggered in this sensitive neuron. So when it comes to evolution and our sensitivity, our visual sensitivity, it's going to depend on the type of creatures that we are. So as mentioned before, birds are going to have higher density of receptors on the top of the eye because they're looking down, right? Rats have a greater density on the bottom of the eye because the things that can kill them are probably coming from above. So let's just real quick review the differences between foveal and peripheral vision. Foveal vision, more cones. When it comes to peripheral vision, you're going to see the proportion of rods to cones increasing. Foveal, one ganglion per cone. When it comes to peripheral vision, each ganglion cell can be excited by many receptors, so rods or cones. When it comes to brightness sensitivity, it's much easier for you to distinguish among bright lights with your foveal vision. You respond very poorly to dim light, but in your periphery, you're going to respond well to dim light, but you're going to have less ease when it comes to distinguishing among bright lights. So you're going to get very good uh, detail vision with your foveal vision, less so with your periphery. Very good color vision with your foveal vision in the focus, but when it comes to the periphery, you're not going to get as great color perception. So we've alluded to this, but now we're going to talk a little bit more in depth. You have two types of receptors in your retina, or rather on your retina. You have rods, which are more abundant in the periphery, as we mentioned before, and they respond more to faint light. You have cones, which are more abundant in and around the fovea, and are more sensitive to specific colors and more useful in bright light. You have about 120 million rods. You have about 6 million cones. So even though there are more rods, cones provide about 90% of the brain's visual input. So there are, on average, 120 million rods, 6 million cones, and they converge onto 1 million axons in the optic nerve. The ratio of rods to cones is higher in species that are more active at dim light. Would you like to guess why? Because rods are more sensitive in dim light. Great job. I'm so proud of you. Here is a rod. Here is a cone. Here are some rods. Here is a cone. That's in your eye. So right now, just pull out your selfie cam, look at those beautiful eyes of yours, and just know in the back of your retina, it looks like that. You're welcome. So photopigments are the thing that allows light to be converted to the chemical signal that our brain uses. So there's something called 11-cis-retinol, and it's bound to proteins called opsins. That light energy that we receive converts 11-cis-retinol into all trans-retinol. So that light is absorbed and energy is released that activates second messengers within the cell, right? We have light, and then we have 11-cis retinol. That turns into all trans retinol, and then it sends signals saying, hey, we've got this color going on. So visible light is a portion of the electromagnetic spectrum. So this includes more than just, the colors that we see. It also includes ultraviolet, infrared, X-rays, radio waves. So those visible wavelengths are dependent on our receptors, what our receptors will respond to. So different species are going to respond to different wavelengths, right? We perceive wavelengths between 400 and 700 nanometers. So now we're going to talk about color vision. There are two major theories when it comes to color vision, the trichromatic theory and the opponent process theory. So the trichromatic theory says that color perception occurs based off of response to different wavelengths of light in different kinds of cones. So some cones are more responsive to short, medium, or long wavelengths. So every cone responds to a broad range of wavelengths, but some are going to be more receptive to certain wavelengths. That ratio of activity across the three types of cones determines the color, right? So if you have three cones, or rather, they should look like that, and these are all super receptive to, let's just say, red light, right? And you got this coming towards it. Well, because those are more responsive to red light, and then you have these other ones over here that are responsive to yellow, right? Then these acting up more, or these sending signals more so than these is going to tell you, oh, we're looking at red. So the more intense that light, that's going to increase the brightness of the color, but doesn't change the ratio. And also, these cones are unevenly distributed. So it's not like, red, yellow, green, blue, right? So here we can see the sensitivity of different types of cones with our shorts over here, medium, long, and then also our rods, right? So if you have something closer to violet, then these short wavelength cones are going to be more responsive, even though they'll still respond a little bit to blue. But if there's red at all, they're not going to be responding at all because that's way outside of their sensitive area. So here's a distribution of cones into different human retinas, and as you can see, they are unevenly distributed, right? So it's not the same, right? You can see these ones that are sensitive to violet, they're pretty sparse, right? You have a lot that are sensitive to red, and just a few that are sensitive to green. Then we have the opponent process theory, which is suggesting that we perceive color in terms of paired opposites. So the according to the opponent process theory, our brain has a mechanism that perceives color on a continuum from red to green and another from yellow to blue. So a mechanism for this theory is that bipolar cells are excited by one set of wavelengths and inhibited by another. So basically, our brain will pay attention to what is firing and what's not firing, and then decide what the color is based off of that. So both of these series have limits. One is the issue of color constancy, which is our ability to recognize color despite changes in lighting. It's not really able to be explained by these two theories. So there's also Retinex theory, which suggests that our cortex, our visual cortices, compare information from various parts of the retina to determine the brightness and color for each area. So when we have context for an image, that's going to affect our perception of the colors in the image. So you might see as you lose more context that the colors seem to change, although they actually are not. There's also what's called brightness constancy, where we kind of have a perception of how bright something is. If you were to put your finger right here and just cover the image and look at it, you might see that the colors of the object shift. And that is because our brains are making assumptions and changing our perception of the image. The sense that we're receiving is still the same, but how our brain interprets what the image looks like is going to shift. Then we have color vision deficiency, sometimes referred to as color blindness. It is often contained on the X chromosome, and by often I mean it is. It's caused by either a lack of the type of cone that would be responsive to that color, or those cones having abnormal properties. The most common form is red green color blindness or color vision deficiency. And it's when the long and medium wavelength cones have the same photopigment. So the interesting thing about vision is it's a physical stimulus that provides a psychological experience, right? There's the actual light that is outside, and then our perception of vision. So we have a pretty detailed understanding of vision, and that actually helps us kind of understand parts of our psychological processes because we're able to look at how our brain is interpreting this info. So just to review, we have rods and cones on the retina. They make synaptic contact with the horizontal and bipolar cells. Horizontal cells in the eye make inhibitory contact with bipolar cells. Bipolar cells make synapses onto amacrine and ganglion cells, and different cells are specialized for different visual functions. Ganglion cells form the axons from those ganglion cells form the optic nerve. The optic chiasm is the place where the optic nerves leaving the eye meet. You might remember that from the diagram. Then you have those little blood vessels. In humans, half of the axons from each eye cross to the other side of the brain. So here's your left eye. One half goes to the left brain. The other half goes to the right brain. So most ganglion cell axons go to the lateral geniculate nucleus. A smaller amount go to the superior colliculus, and they also go to a few other areas. Here's another view of the retina. We have our rods and cones, horizontal and bipolar cells, amacrine cells, and ganglion cells. And here's another view right here. So we have our retina, the optic chiasm, we have our optic nerves, and then we have the lateral geniculate nucleus, our superior colliculus, and our visual cortices in the back of the occipital lobe. And then you can also see here the optic chiasm where the cross happens, right? So I have the left side of my left eye, the right side of my right eye, and then the left side of my right eye, and the right side of my right eye. So let's talk real quickly about lateral inhibition in the retina. What this does is it sharpens contrast to emphasize the borders of objects, right? So you have this square, right? Uh, there's this contrast between the blue and the white, and so you can see this as a separate object, right? And if it had like a little bit of a shadow to it, then you would see, oh yeah, that's definitely popping from the background, right? Or you'd go, Professor Thompson's not good at drawing. Either way, the reduction of activity in one neuron occurs from activity in neighboring neurons. So the combination of responses in cells in your visual system is going to depend on what is sending excitatory signals and inhibitory signals, right? So by looking at everything, our brain is able to really understand what is in the foreground, what is in the background, and have a better, more thorough understanding of the world around us. So here's an example of your brain trying to make sense of the world. You might notice at the little intersections that there are some gray dots that seem to appear and not appear. It's because I put LSD in your drink. You're tripping balls. No, it's not drug induced This is because our brain is trying to make sense of information, and in trying to do so, right, we're shifting our focus. So maybe you're looking over here, and then you're looking over here, and in the periphery around your fovea, your brain's trying to make sense of these little crosses, right? So it's filling in information, which is making these little gray dots appear. Oh, now it's gone. Right there. Nope. Now it's actually. Oh, wait. Nope. It's here now. Ah. So this processing occurs in the lateral geniculate nucleus. It's part of the thalamus that is specialized for visual perception. It's where most of your ganglion cell axons end up. So it also sends to different parts of the thalamus and to the visual areas of your occipital cortex. The cortex and thalamus are constantly feeding information back and forth to each other. They are two good friends. They chisme all the time. They're just like, oh, did you see what's moving there? Oh, yeah, I saw that. Did you notice the color on that thing? Yeah, I noticed the color on that thing. They're the best of friends. They have a great time. So our receptive field is the part of our visual field that is exciting or inhibiting cells in the visual system of the brain. So for a receptor, it's the point in space from which light strikes that receptor. So if you're looking at this tree, it's a Christmas tree. It's very lovely. So from right here, right, light bounced off from I guess the sun or something, and then hit your eye. Well, technically, it switches because of the lens. So that is the receptive field. For other cells, receptive fields are derived from the visual field of cells that either excite or inhibit, right? So, for example, you have your ganglion cells that converge to form the receptive field for the next level of cells. So even though the receptor is only responsive to one point of light, the ganglion cells, because they're getting input from multiple receptors, have a wider receptive field because one receptor might be saying, oh, there's light right here, but all the other ones are saying, oh, there's nothing. So these multiple receptors increase the wideness of the field because they're all sending yes or no signals. There are three categories of ganglion cells when it comes to primates. You have parvocellular, magnocellular, and coniocellular. Parvocellular neurons are mostly located in or near the fovea. They have smaller cell bodies and small receptive fields, and they are highly sensitive to detecting color and visual detail. Magnocellular neurons are distributed evenly through the retina. They have larger cell bodies and visual fields, and they're very sensitive to large overall patterns and moving stimuli. And then coniocellular neurons have small cell bodies. They're found throughout the retina, and they have several functions, and their axons will terminate in many different places. So when it comes to receptive fields, the cells of the lateral geniculate nucleus have a receptive field similar to those of ganglion cells. So they have an excitatory or inhibitory central portion and a surrounding ring of the opposite effect. So they're maybe sensitive here to excitatory stimuli, and then they're surrounded by a ring of inhibitory stimuli. When it comes to the primary visual cortex, area V1 receives information from the lateral geniculate nucleus and is responsible for the first stage of visual processing. If you are damaged in your V1, you might experience what's called blind sight, where you're able to respond to visual stimuli that you don't report seeing. So somebody throws a ball at you, and then you like duck out of the way, right? You might not be able to say that you saw the ball, but your brain perceived it. So that's what would happen if you experience damage in the primary visual cortex. So in 1959 and 1998, Hubel and Weissel distinguished various types of cells in the visual cortex. So we have simple and complex cells, and then we also have the end-stopped or hypercomplex cells. So simple cells have fixed excitatory and inhibitory zones. The more light that shines in the excitatory zone, as you might imagine, the more the cell will respond. The more in the inhibitory zone, interestingly enough, the less the cell responds. So they have bar-shaped or edge-shaped receptive fields with vertical and horizontal orientations that outnumber diagonal ones. So they're more responsive to light that looks like that, right? Complex cells are located in either V1 or V2. They have a large receptive field that cannot be mapped into fixed excitatory or inhibitory zones, and they respond to patterns of light in a particular orientation. So they're very receptive to when light is arranged in a specific way, and they respond most strongly to a moving stimulus. So let's say you have a vertical line and you have some simple or complex cells that are more responsive to vertical lines. If that line were to tilt and be horizontal, you wouldn't really have much of a response. Once it gets close to that, sorry, let me make it a little bit more slanted, then it's going to have a stronger response. But over here, when it's perfectly vertical, then you're going to get the strongest response because it's responsive to vertical stimuli. So what you saw previously was light that was in a particular orientation. So that simple cell best responds to light that's in that particular place, in that particular orientation, right? Right in the middle of the visual field, perfectly vertical. Now, a complex cell can respond regardless of where it is in the visual field. So we can see that this complex cell is responsive to diagonal orientations, and regardless of where it is in the visual field, you're going to get a high response, right? But once it becomes more vertical or more horizontal, you're getting a lower response. So simple cells needed to be right in a particular place, right, in a particular orientation. Complex cells, the location where it is doesn't matter as much, but the orientation still matters. Then we have end-stopped or hypercomplex cells. These are similar to complex cells, but they have a strong inhibitory area at one end of the bar-shaped receptive field. These can respond to a bar-shaped pattern of light anywhere in its receptive field, provided that the bar doesn't extend past a certain point. So these all have a strong response, but once that bar crosses this end stop, then there's a weak or no response. So to summarize, simple cells are located in V1, complex and end-stopped cells are in V1 and V2. Both have, or all three, sorry, have binocular input. The receptive field is smallest in simple cells, largest in end-stop cells, and halfway between the two in complex cells. The receptive field in simple cells is bar or edge-shaped with very fixed excitatory and inhibitory zones. In complex cells, it is edge-shaped, but it responds equally through a large receptive field. And in end-stop cells, it's the same as complex cells, but with that really strong inhibitory zone. So in our visual cortices, cells are grouped together in columns that are perpendicular to the surface. Cells within a given column process similar information. They're either going to respond mostly to the left or right eye, or they'll respond to both eyes equally, and they do not consistently fire at the same time. So if you were to put an electrode through someone's brain, if you just put it through one column, you'll see that you're hitting cells that are responding to the same orientation. If you were to cross through different columns, as you're doing here and here, you're going to hit cells that are responding to different orientations. Some cells in the visual cortex are feature detectors, meaning that they respond to a particular feature or stimuli. When you are exposing those cells to a given feature over time, the sensitivity to that feature decreases. So we're very responsive to faces. If you look at the front or rear of a car, your brain sometimes goes, "Oh, look, the car has a little face," right? That's because our brain is designed to find faces in the world easily, right? That is a safety mechanism. If you see a face, you can find your friend in a crowd, or if you're out in the wild, you can find a predator hopefully before it finds you. Ooh, scary. So, by studying animals, we've learned a lot about how vision develops. Early lack of stimulation in one eye leads to those synapses in the visual cortex becoming gradually unresponsive to input from that eye. And early lack of stimulation in both eyes makes our cortical responses sluggish, but doesn't cause blindness. So, that old adage, use it or lose it, right? If you're not getting stimulation, then you're going to see a lack of the proper development of those pathways. So when it comes to development, you may have heard the term sensitive or critical periods. Sensitive periods are when there's like a best time to learn something. Critical periods are when you have to learn something by a specific point or you'll never learn it. So if you think of learning a new language, much easier to do when you're younger. There's a sensitive period for learning language, but if right now you're like, oh, I want to learn Russian, then you could still learn it. It's just going to be much more difficult. So when it comes to vision, this is ending with the onset of chemicals that inhibit axonal sprouting. So the more pathways that you can create, the more likely you are to be able to forge those pathways that allow you to perceive things in the proper way. So our cortical plasticity is greatest early on in life, but it never ends. You might know that depth perception is based off of the fact that we have two eyes. This relies on our retinal disparity. So basically, there's a slight difference between what our left and right eye sees. And by putting those images together, we can determine how far away something is. Strabismus, also known as having a lazy eye, often develops in childhood. This is when the eyes don't point in the same direction. What can happen is if your two eyes are carrying unrelated messages. So, we just talked about stereoscopic depth perception, where we're comparing the two images. But if you're getting unrelated information, what might happen is your cells will only strengthen connections with one eye uh, and only trust or primarily rely on messages from one eye, meaning that your development of depth perception is impaired. And here's a example of a lazy eye. So, if you're only exposed to a limited array of patterns, this is going to cause almost all of your visual cortex cells to become responsive only to that pattern. So, astigmatism, which you might have heard of, refers to a blurring of vision for lines in one direction caused by an asymmetric curvature of the eyes. 70% of infants have astigmatism. So I know what you're wondering. How do you test how to restrict early visual experience? Is it by putting cats in what look like VR goggles and a cone? Why, yes, it is by putting little kittens in these little goggles, which only give a very narrow like visual experience. So this would be per eye, and then the rest of the time that they are alive, they are in a dark room with their mom. So they're getting very little visual exposure aside from the controlled visual exposure that they see, which leads to their pathways only being responsive or being primarily responsive to, let's just say in this case, vertical stimuli. So here's a little informal test for astigmatism. So, do the lines in one direction look darker or sharper than the other lines do? If so, see if something happens when you rotate the page. If you're wearing corrective lenses, contacts, glasses, etc., try it without or with your lenses. If you got laser eye surgery, then take some lasers and no, actually, no, not gonna, not gonna go there. No lasers towards your eyes. So, in studies of people with cataracts, having them removed can help restore vision gradually, but problems will still persist. These problems can include difficulty in recognizing objects or being able to recognize components as part of a whole. The best prognosis for any visual problems comes from early diagnosis and treatment. So we've identified many different parts of the brain that contribute to vision in different ways. We have the parts that see shape, the others that see color, another area for detecting of location, and also for movement. So the secondary visual cortex, area V2, receives information from area V1, processes that information further, and sends it to other areas. Information is transferred between V1 and V2 in a reciprocal nature. So there are ventral and dorsal streams. So remember our dolphin. Such a good dolphin. Probably the best dolphin that's ever been drawn in the world. So the ventral stream refers to the path that goes through the temporal cortex. This is the what path. It helps us identify and recognize objects. The dorsal stream refers to the visual path in the parietal cortex, which helps us understand how. So, how is this moving, and where might it be going? So we're never using these separately. We're often using both of these in collaboration. So any damage to either stream will produce different deficits. If you get damage to the ventral stream, you can see where objects are, but you're going to have trouble identifying them. If you experience damage to the dorsal stream, you'll be able to identify objects but not know where they are. Our receptive fields become larger and more specialized as visual information goes from the simple cells to the complex cells and then to other brain areas. Our inferior temporal cortex contains cells that respond selectively to complex shapes but are insensitive to the distinctions that are critical to other cells. So, those temporal cells really help us understand. So, for example, you see like a star, right? The this type of thing. It's pretty complex, especially if it's as greatly drawn as that one is. But being able to understand that complex shape would be the job of the inferior temporal cortex. So the cells in that cortex respond to identifiable objects. So you know that's a star. You've seen it before, so you can very quickly go, that is a star. So how an object is transformed is going to affect our perception of it. Here's the original, and we can see the face. If we flip it, then it still feels like a face to us. If we change the colors, so if we switch the contrast, then it still feels like the same image. Now, there's something called figure ground, which is the balance between what is the foreground and what is the background. So if we change the background, then our brains will try to focus on this white part. And so our brains start to go, wait, what's the thing that we're supposed to be focused on, right? So if you try to look at that image, you might see that for a second, your brain focuses on the white and it looks like some, sort of like crown molding on, a wall or ceiling or something like that. And then maybe you pop back in and see the face, right? It's much more difficult to perceive just the face by itself. There's also what's called visual agnosia, which is your inability to recognize objects even though your vision is working. And this is caused by damage to the pattern pathway, which is usually in the temporal cortex. So if I do this, right, what am I drawing? Uh-oh, look at it. Look at it. What is it? It's a cat, hopefully. So now it's fading away, right? If you have visual agnosia, then the light is bouncing off that cat and hitting your retina, but your brain is not able to recognize it. You can see it, you can't recognize it. Face recognition happens here in the fusiform gyrus. When you think fusiform gyrus, which I know you think about a lot, think facial recognition. Face recognition is one of the earlier things that we learn. Newborn babies show a pretty strong preference for a properly oriented face, right? If it were upside down, the newborn would not recognize it as well. And then we continue to develop that sense gradually into adolescence. So, in a study of the amount of time that infants spent looking at specific patterns. They spent the most time focusing on faces. They also like circles. They like newsprint. The color red is the least appealing. So we can see that they have an inborn preference for faces. We could also see how they prefer certain orientations of faces. So in this case, they prefer the upright face over the upside down face. They prefer a top heavy configuration face, meaning all the parts are where they should be, but they're oriented weird, versus a bottom heavy configuration. And then, interestingly enough, they share an about equal preference for an upright face or top heavy configuration. So the focus is on, are all the parts where I expect them to be? And if so, this is the thing I'm going to focus on, even if they're not oriented how they should be. Prosopagnosia is the impaired ability to recognize faces. If you've ever heard of the book The Man Who Mistook His Wife for a Hat by Oliver Sacks, it talks about various types of visual impairment. One of them is an example of prosopagnosia. This can occur after damage to the fusiform gyrus of the inferior temporal cortex, which we talked about. The fusiform gyrus responds more strongly to faces than anything else. So then, when we're talking about motion perception, uh, all four lobes of our cerebral cortex are involved. So we have the middle temporal cortex, the MTB5, that responds to stimuli that are moving in a particular direction. Then we also have cells in the dorsal part of the medial superior temporal cortex, the MST. This responds to any expansion, contraction, or rotation of a visual stimulus. And then both of these receive input from the magnocellular path, and they are color insensitive. So for this pathway, color does not matter. So things that are exciting to the dorsal part of the area MST, expansion, so seeing things get larger, and also rotation when there's any movement in the orientation of something. There's also what's called motion blindness, which is an inability to determine the direction or speed of objects or whether or not something is moving. This is likely caused by damage to that area in the MT. Some people are blind, except for the ability to detect which direction something is moving. So that means that the area MT probably still gets some visual input, even though there's damage in the V1 area, which is where most visual information is being primarily routed to. So saccades are little twitchy like movements of the eye. They're a natural thing that our eyes do. And if you think about it, like if you're holding a camera and your hands are shaky, the video is going to come through kind of like blurry, or it might be kind of unclear what's happening. So even though our eyes are making these little movements, we're still able to have a kind of clear, and by kind of clear, I mean very clear, picture of what's happening in the world. So, what's happening during a saccade, there's a decrease in the activity of the visual cortex during those movements. Neural activity and blood flow decrease 75 milliseconds before and during those eye movements. So, the brain is basically compensating for those saccades. So that is visual processing. Now you know what you see. So there we go. We did it. Good job. Take a break. Drink some water.